Welcome to part 3 of the lectures for the urinary system. Today we are finishing up the last of these lectures, and we're starting with looking and discussing the transport mechanisms that are involved for the kidneys. So solute reabsorption is going to drive water reabsorption based on osmosis. So the mechanisms that are going to accomplish sodium reabsorption in each portion of the renal tubule and the collecting duct are going to recover not only filtered sodium, but also other electrolytes and nutrients in water. So the reabsorption of sodium by the renal tubules is going to be particularly important, and this is because of the large number of sodium ions that are going to pass through the glomerular filter. We're going to have a couple of different types of transport that can be used for this. So just a brief reminder, primary active transport is going to make use of ATP in order to power a sodium-potassium pump. Right? So at rest, this is going to account for about 6% of the body's total ATP use. Then we have our secondary active transport, and this is going to make use of the ion's electrochemical gradient in order to drive the transport of these various solutes. So symporters are going to move substances in the same direction, and antiporters are going to move substances in opposite directions. Solute reabsorption is going to drive water reabsorption, and this is because all water reabsorption is going to occur via osmosis. So about 90% of the reabsorption of water that's filtered by the kidneys is going to occur along with the reabsorption of solutions such as sodium chloride and glucose. Water reabsorption with solutes in the tubular fluids is going to be termed obligatory water reabsorption, and this is because water is obliged to follow the solutes when they are reabsorbed. This type of water reabsorption is going to occur in the proximal convoluted tubules as well as in the descending limb of the nephron loop. And this is because these segments of the nephron are always going to be permeable to water. Reabsorption of the final 10% of water, in total about 10 to 20 liters per day, is going to be termed facilitative water reabsorption. And it's facilitative because it's going to be capable of adapting to a need, right? So facilitative water reabsorption is going to be regulated by antidiuretic hormone and is going to occur primarily in the collecting ducts. We're then moving forward to the reabsorption and secretion. So following that filtered fluid from the proximal convoluted tubule into the nephron loop and then on into the distal convoluted tubule and then through the collecting ducts, the filtered fluid from our tube is going to become tubular fluid once it enters that proximal convoluted tubule. And the composition of the tubular fluid is going to change as it flows along that nephron tube and through the collecting ducts. And this is due largely to the reabsorption and secretion of various solutes. The fluid that drains from the papillary ducts into the renal pelvis is what is going to be ultimately known as urine. So looking at the reabsorption that's going to occur in the proximal convoluted tubule, most of the solute reabsorption in this proximal convoluted tubule is going to involve sodium. And so sodium transport is going to occur via symport and antiport mechanisms in the proximal convoluted tubule. And normally filtered glucose as well as amino acids, lactic acid, any kind of water-soluble vitamins, and some other nutrients are not going to be lost in the urine. They are going to be completely reabsorbed in the first half of the proximal convoluted tubule. And this is going to occur via several different types of sodium symporters that are going to be located in the apical membrane. So two sodium molecules and a molecule of glucose are going to be attached to the symporter protein, and this is going to then carry them from that tubular fluid into the tubule cell. The glucose molecule are then going to exit the basolateral membrane via facilitated diffusion, and they're going to diffuse into the peritubular capillaries. The other sodium symporters that are found in the proximal convoluted tubule are going to be involved in reclaiming filtered phosphate, as well as sulfate ions, as well as any amino acids and lactic acid that might be present. And this is going to all happen in a very similar way. Next, we have the reabsorption and secretion that's going to take place in the proximal convoluted tubule. So, in another secondary active transport process, the sodium-hydrogen antiporter system is going to carry filtered sodium down its concentration gradient and into the proximal convoluted tubule cells. As those hydrogen ions are going to be moved from the cytosol into the lumen. This causes the sodium to be reabsorbed into the blood, and the hydrogen can then be secreted into the tubular fluids. The proximal convoluting tubule cells are then going to produce the hydrogen that is needed to keep those antiporters running in the following way. So CO2, carbon dioxide, is going to diffuse from the peritubular blood or from the tubular fluid, or sometimes also is going to be produced by metabolic reactions that are happening within the cell. It can also occur in red blood cells, where the enzyme carbonic anhydrase is going to catalyze the reaction of CO2 with water to form that carbonic acid. And this is going to then dissociate to form hydrogen ions and bicarb. So then moving forward to the reabsorption and secretion that we see, continuing with the reabsorption and secretion in the proximal convoluted tubule, most of the bicarbonate ions that are being filtered, that are in that filtered fluid, are going to be reabsorbed in the proximal convoluted tubule, and are thereby going to help to safeguard the body's supply of this really important buffer, because bicarb is a very important buffer for the body. So after hydrogen is secreted into the fluid within the lumen of the proximal convoluted tubule, it's going to react with filtered bicarb in order to form carbonic acid. This is going to readily then dissociate into CO2 and water. The carbon dioxide is then going to diffuse into the tubule cells and is going to join with the water to form the carbonic acid, which is then ultimately going to dissociate to produce hydrogen ions and bicarbonate. So as our levels of bicarb increase in the cytosol, it's going to exit via facilitated diffusion via those transporters in the basolateral membrane, and it's also going to diffuse into the blood with sodium. Therefore, for every hydrogen ion that's secreted into the tubular fluid of the proximal convoluted tubule, there's going to be one bicarb ion and one sodium ion that are reabsorbed. And then moving forward, as we move on down our nephron here, we're moving on to the passive reabsorption in the late proximal convoluted tubule. Right, so the solute reabsorption in this proximal convoluted tubule is going to promotes osmosis of water, and each reabsorbed solute is going to therefore increase the osmolarity, first inside the tubule cell, and then within the interstitial fluid, and then finally within the blood. So then water is going to move, therefore, more rapidly from the tubule fluid via both the pericellular and transcellular routes. And it's moving into the peritubular capillaries and is going to help to restore osmotic balance. So essentially, the reabsorption of the solutes is going to create an osmotic gradient that is therefore going to promote the reabsorption of water via osmosis. So cells that line the proximal convoluted tubules and the descending limb of the nephron loop are going to be especially permeable to water, and this is because they're going to have many molecules of aquaporin 1. This is a really important integral protein that's found in the plasma membrane of these areas and is going to be essentially a water channel. It's going to really help increase the rate of water movement across the apical and basolateral membranes. That brings us into the reabsorption that's occurring in the loop of Henle. So the nephron loop is going to set the stage for the independent regulation of both the volume and osmolarity of the body's fluids. So sodium, potassium, and chloride symporters are going to reclaim those ions from the tubular lumen fluid. And because potassium leakage channels are going to return much of that potassium back into that tubular fluid, the main effect of these sodium, potassium, chloride symporters is going to be the reabsorption of sodium and chloride. So although about 15% of the filtered water is going to be reabsorbed in the descending limb, there's going to be little to no water is actually going to be reabsorbed in the ascending limb. So then moving onward, we have the reabsorption that's going to occur in the early distal convoluted tubule. So as fluid flows along the distal convoluted tubule, the reabsorption of sodium and chloride is going to continue due again to those to the sodium chloride symporters. And the distal convoluted tubule is going to serve as a major site where parathyroid hormone is going to stimulate the reabsorption of calcium. So it's also going to be really important at inhibiting phosphate reabsorption that's found in the proximal convoluted tubule, and this is going to enhance its secretion. So the solutions are going to be reabsorbed with very little accompanying water. So then moving on to the late distal convoluted tubule and its associated, and then the following collecting ducts. Sodium is going to pass through the apical membrane of those principal cells, and it does so via a sodium leakage channel. The sodium pumps are actively going to transport sodium across that basolateral membrane. Then the secretion of potassium through our potassium leak channels in the principal cells is going to be the main source of potassium that's going to get excreted into the urine. Then finally, our principal cells are also going to secrete a variable amount of potassium. So because of the basolateral sodium-potassium pumps continually bringing potassium into those principal cells, potassium is going to remain high. So our potassium leakage channels are also going to be present, and they are going to allow potassium to diffuse down its concentration gradient into that tubular fluid. So moving forward, looking at the homeostatic regulation of the tubular reabsorption and secretion that's going to go on throughout the kidneys. So there are going to be four main hormones that are going to impact the extent to which sodium, chloride, and water are reabsorbed by the kidneys and how much potassium is being secreted by those renal tubules. Right? These hormones are angiotensin II. This is going to be part of the renin, angiotensin, and aldosterone system. And this angiotensin II is going to increase blood volume as well as blood pressure, and is going to be a major regulator in electrolyte reabsorption and secretion along with aldosterone. So these two are going to work together because they're all part of that renin, angiotensin, that RAA pathway that we talked about. So all this together is going to help to increase the reabsorption of water in those collecting ducts. All right, so our other three hormones are antidiuretic hormone, atrial natriuretic peptide, and parathyroid hormone. All of these are going to be really important at regulating the reabsorption of sodium, chloride, and water, and the secretion of potassium. So starting with antidiuretic hormone, ADH, this is going to regulate facilitated water reabsorption by increasing the water permeability of those principal cells. Next, our atrial natriuretic hormone, or peptide, This is going to inhibit both water and electrolyte reabsorption. And then last but not least, our parathyroid hormone. This is a really important one in promoting calcium reabsorption. So then looking at the regulation of water reabsorption and how this is impacted by ADH. So as with so many other things in the body, this is going to be controlled by a negative feedback loop that involves ADH regulating the facilitative water reabsorption. So when the osmolarity or osmotic pressure of plasma and interstitial fluid increases, i.e. when water concentration decreases, by as little as even 1%, the osmoreceptors that are in the hypothalamus are going to detect this change. Their nerve impulses are then going to stimulate the secretion of ADH into the blood, and the principal cells are going to become more permeable to water. So as facilitative water reabsorption increases, blood or plasma osmolarity is going to therefore decrease to a normal level. So a second very powerful stimulus for ADH secretion is going to be a decrease in blood volume, the kind that would occur if you were to have like a massive hemorrhage or some sort of very severe dehydration. This would also result in this secretion of ADH from the So, moving forward then to look at how angiotensin II is going to impact renal physiology, there are three primary ways here. First of all, it's going to decrease the glomerular filtration rate by causing vasoconstriction of those afferent arterioles. All right. It's also going to enhance reabsorption of sodium and water in the proximal convoluted tubule, and it does this by stimulating the activity of sodium-hydrogen antiporters. It also is going to stimulate the adrenal cortex in order to release aldosterone, and this is going to be a hormone that's going to in turn stimulate those principal cells in the collecting ducts to reabsorb more sodium and secrete more potassium. The osmotic consequence of reabsorbing more sodium is going to be that more water is going to be reabsorbed, which in turn is going to cause an increase in blood volume and blood pressure. So here we have a nice little table for your studying convenience looking at the various hormone regulations that are going to occur in the tubular reabsorption and tubular secretion. So, moving forward now to look at the production of urine. All right, so fluid intake is going to be highly variable for most people. Right? And homeostasis is going to therefore require maintenance of fluid volumes within specific limits. So urine concentration is going to vary with ADH levels. And high intakes of fluid is going to result therefore in a dilute urine of high volume. And alternatively then, low intake of fluid is going to result in concentrated urine of low volume. So this is why if you're one of those people who's drinking like a gallon of water a day, you're going to have to tinkle all the time, and your urine is going to be very light in color because it's very dilute. On the other side of that, if you're one of those people who has like a sip of water in the morning and then nothing until you go to bed, you're going to pee less, but you're going to have very dark colored urine because this stuff is going to be way more concentrated. So, fun facts there. You can learn a lot from your pee. So, let's start then with the formation of dilute urine specifically. So, there are going to be about 6 steps that are involved with this. So, in step 1, when ADH levels are very low, the kidneys are going to produce dilute urine and are going to excrete that excess water. So, in other words, those renal tubules are going to absorb more solutes than they are absorbing water. So, although solutes are being reabsorbed in the thick ascending limb, the water permeability of this portion of the nephron is always going to be quite low. And this is going to allow water to not be able to be followed by osmosis. Right? So then the fluid that's entering the distal convoluted tubule is going to be more dilute than what we would have in the blood plasma. So the early distal convoluted tubule cells are not going to be very permeable to water and are therefore not going to be regulated by ADH. Finally, the principal cells of the late distal convoluted tubules, as well as those collecting ducts, are going to be impermeable to water when the ADH levels are very low. Therefore, the tubular fluid is going to become progressively more dilute as it's going to flow onward. So by the time that tubular fluid drains into the renal pelvis, its concentration can be as low as 65 to 70 milliosmoles per liter. So very dilute is what that essentially the take-home message is there. So when you're forming this dilute urine, the glomerular filtrate and blood is going to have the same osmolarity. All right, so it's going to be about 300 milliosmoles per liter. However, it's important to remember that the tubular osmolarity is going to change due to a concentration gradient that's found in the medulla. So when dilute urine forms, the osmolarity that's in the tubule is going to increase in the descending limbs. It's going to decrease in the ascending limbs, and it's going to decrease even further in the collecting tubules. So, in the thick ascending limb, when we're forming this dilute urine, the symporters are actively going to reabsorb sodium, potassium, and chloride, and it's going to have a very low water permeability. Also, solutes are going to leave, and water is going to stay in the tubules. This is why it's so dilute. Then within the collecting ducts, there's also going to have low water permeability, and this is due to that absence of ADH. So hello. Sorry about that, I had a little technical difficulties. So as far as the formation of concentrated urine is concerned, when ADH levels are high, the kidney is going to secrete concentrated urine in order to conserve water. So a large volume of water is going to be reabsorbed from that tubular fluid into the interstitial fluids, right? And the solute concentration of the urine is also going to be high. So the production of concentrated urine is going to involve the ascending limb of the nephron loops, and this is going to be really important in establishing the osmotic gradient that we see in the renal medulla, as well as in the collecting ducts, reabsorbing more water and more urea. And the urea cycling is also going to cause a buildup of urea in that renal medulla. So, for this formation of concentrated urine, the juxtaglomerular is going to happen primarily in the juxtamedullary nephrons with the long loops. The osmotic gradient is going to be created by the countercurrent multiplier, which we're going to talk about in just a second. Solutes are going to be pumped out of the ascending limb. However, water is going to stay in those tubules, and the medulla osmolarity is going to increase. So in the presence of ADH, the collecting ducts are going to become very permeable to water, and that tubular fluid here is going to become very concentrated. So the movement of water is also going to carry urea into the medulla, and this is going to help also contribute to its osmolarity. So now let's look at that countercurrent multiplication that we looked at, or that we mentioned. So the countercurrent mechanism is also going to contribute to the excretion of concentrated urine. So countercurrent multiplication is going to involve the long loop of the loop of Henle of those juxtamedullary nephrons, which we see here. Right. So symporters in the thick ascending limb cells of the nephron loop are going to cause a buildup of sodium and chloride in the renal medulla, and countercurrent flow through the descending limb and the ascending limbs of the nephron loops are going to help to establish an osmotic gradient in that renal medulla. So cells in the collecting duct are going to reabsorb more water and more urea, and the urea recycling is going to cause a buildup of urea in that renal medulla. So because of this, we have our countercurrent exchange in this solutes. This is where solutes and water are going to be passively exchanged between the blood of the vasa recta and the interstitial fluid of the renal medulla, and this is a result of countercurrent flow. So with this countercurrent exchange, the loop and duct cells are going to require nutrients and oxygen from the blood supply. So capillaries that feed them, i.e. the vasa recta, are going to form loops like those that we see in the nephron loops of the medulla. And so incoming and outgoing blood will have a similar osmolarity. And this is going to be really important in maintaining the medulla concentration gradient. So once we've produced all of this urine, be it concentrated or dilute, we have to transport it and store it somewhere. Otherwise, we'd all be just kind of walking around sort of leaking all the time, which would be rather inconvenient, I suppose. So for this, each ureter is going to transport urine from the renal pelvis to in peristaltic waves using hydrostatic pressure and gravity. So there are no anatomical valves at the opening of the ureter into the bladder. All right, this means that they're going to be once you're dead, essentially. If you're, there's not going to be any ring of muscle that's specifically dedicated to that. All right. So when the bladder fills, it's going to compress the openings, and this is going to prevent backflow. The bladder is going to be a hollow distendable muscular organ, and it's going to have a capacity between 700 and 800 mils. So you can hold a pretty good bit of urine in there, but it's important. So that brings us to something called micturition. This is going to be the discharge of urine, and it's going to involve voluntary and involuntary muscle contractions. Stretch receptors are going to trigger a spinal reflex, which we are going to have control of or gain control of in early childhood. This is why you're not born knowing how to hold it, essentially. The urethra is then going to carry urine from the internal urethral orifice into the exterior of the body. And in males, this is also going to be responsible for the discharge of semen as well as urine. So a little bit of a difference there. So that is all that we have to cover for the urinary system lectures. That is our last one of the whole year. So as always, if you have any questions, comments, or concerns, please feel free to contact me or ask after class. So good luck.